Okay, so the first question that was
Mhm.
in the chat during the talks was, is there a blood test for the inflammation caused by MSA?
Uh, okay, yeah, that's an interesting question. So the short answer to that question is no. So there is no blood test or no specific blood test that could be used for diagnosis of multiple system atrophy. But if we look at inflammation or inflammatory markers, we do see that changes in inflammatory markers in the blood do correspond with those that, for instance, we see in the brain. But these are very generic responses, and so very often these are also seen in other diseases like Alzheimer's, other types of dementias, PSP, and Parkinson's disease. So it's very difficult just to distinguish and differentiate these neuroinflammatory changes in the blood and use them as an actual diagnostic marker of multiple system atrophy. So there are definitely changes, and they are observed in the blood, um, but it's difficult to use these to say anything convincingly about MSA itself.
And some questions now that were posted in the chat just now. My question is regarding the facilitators, the effective process of inflammation to MSA. What facilitators have been studied?
Also a good question. So there have been a couple of facilitators that have been studied, especially genetic risk factors. And so recently, I think there was a study published, a genome-wide association study looking at risk genes or risk variants that might associate with multiple system atrophy. And if I'm not mistaken, I think that study included over 700 patients with multiple system atrophy. But eventually the study was negative, so they couldn't identify a significant risk factor, genetic risk factor that could serve as a facilitator. There were a couple of hits that were just below, I think, significance threshold, meaning that maybe some genes might be associated or could act as this facilitator that I was talking about, but that the population should be expanded in order for these genes to become significant. And so a facilitator could be a gene, but so far all of these genetic studies have come up negative, which either means that we need more people to test or to look for genetic risk factors or genetic facilitators, or simply that a genetic component in MSA is just really weak. And so it seems that that both are probably the case. As in other facilitators, in that sense, I'm thinking about maybe inflammatory conditions, other infections, autoimmune conditions, even maybe even somatic genetic mutations and so forth. So there could be several facilitators that could lead to the development of multiple system atrophy after a certain trigger might occur in the infectious hypothesis, and of course. But so far we haven't found them, or we haven't discovered what those could be.
Okay, thank you. Dr. Espay, I have a question for you. So in your talk, you emphasized the idea of approaching increasing the healthy proteins as opposed to focusing so much on the abnormal proteins. And you use the analogy of a fire and looking at the ashes of a fire. I wonder though, if an analogy of a clogged sink might work here too, where you have a clogged sink, it's full of these abnormal things they're clogging up the water system, but adding more water to the system won't necessarily improve the process. So what is the evidence that your approach could actually work in addressing the problem?
Yeah, great question. And I like your analogy. Right. Part of the interest in continuing to work on proteins as toxic in the research field is because it is what looks abnormal in a brain under a microscope after somebody has died. So if it's abnormal, the question is it the cause of the problem, right? And part of the reason I use a bit of the ashes of the fire, because that analogy implies we're really looking at somebody's tissue after death. And we're trying to make sense of what happened during life, literally on the ashes of the fire. So that's part of the reason why that analogy is a little bit more appropriate than the clogged water sink that you're saying. But here's why we began to start looking at this in a manner that would start sort of thinking that perhaps we're really looking at the victim, not at the villain. And that is that we were trying to get from human studies, what is the relationship between the aggregated burden and distribution of these proteins and degeneration or behaviors, abnormal symptoms, the symptoms that would define diseases. And we found that there isn't a relationship, right? And so if you would expect that this is really what we need to get rid of, then there must be at least some kind of correlation between how much of the pathology, the abnormal proteins there are, and how severe a disease is, right? But there isn't such a relationship. And in clinical pathologic autopsy studies that have been done now, there is a great paradox, which is that most people with these abnormal proteins had no symptoms during life. So while there is a correlation between the onset of these pathologies and later on the development of what we would refer to diseases from the pathologies, a lot more people with pathologies that do not develop any of the symptoms than people with pathology that do. So therefore, something is missing. So in MSA, we don't have a way to study this in life, in humans. So we just do animal models. And if in animal models, we inject a protein in any way, give animals more of this protein, they get sick. And if we remove it, they get better. So in animal models, it's easier to speak of causality of these pathologies, these proteins causing the disease. In humans, it's a lot complicated because while it is true that the pathology is there before the disease, so that's one criteria met, for a demonstration of A causing B in humans, we need 9 criteria. And all the others aren't met. And one important one is that the more of A, the cause, the more of B, the disease. And that isn't the case. So part of what I'm trying to emphasize with my presentation is trying to remind us that abnormal proteins were once normal. And that part is being invisible in research, completely invisible. In fact, if you look at the presentation, yeah, go ahead.
Yeah, no, I just want to clarify something you said a minute ago. Are you saying that there have been studies done on brain tissue that have found the same types of misfolded proteins that were seen in MSA, but those patients didn't have symptoms? Because right now, the only way you can diagnose someone, except on autopsy with MSA, is through symptoms. So are you saying that there have been brains that have been found to have the same types of infections or not infections, but accumulations of these misfolded proteins, but that those patients had never been diagnosed with MSA?
Correct. That's what Andrew Lees called the dirty little secret. And I think of it as not a little secret. This is a big secret if we looked at the fact that, in fact, in brain banks, all of them, there are more people with pathology than people who had history of disease. In Parkinson's, it's interesting that this was a collection of 110 brains, thereabouts. Only 40 were brains of people who had Parkinson's. The rest were people without Parkinson's with the pathology. So all brain banks have a lot more people with the pathology that never had neurological symptoms compared to people with pathology and neurological symptoms. So that shouldn't be a secret, but it is something that we've chosen to neglect. I think in general, in a narrative where there is a villain, we like it, we're attracted to it, we think is a powerful narrative. And let's face it, we love to galvanize against a common enemy. The trouble is that in other fields now, not in MSA necessarily yet, although we're moving in that direction, we've been really good at eliminating what we have considered the villain. We have 2 decades of that approach already in Alzheimer's, and we're accumulating quite a bit of it in Parkinson's. Nothing to show for it. Now, the studies have accomplished what we have wanted to. We've been able to clean out the protein. So we would be able to eliminate or reduce the protein that accumulates in Alzheimer's. But the problem is that what we're missing is the normal protein. That component is invisible. We, in many ways, have created the idea that these abnormal clumps of proteins emerged by spontaneous generation. No, they were the tombstones, or are the tombstones of proteins that once were normal, and the only opportunity for proteins to function is when they are normal. The minute they turn into something they aren't, they cannot function.
Okay, so I don't want to monopolize the whole time with this topic. Wouter, do you have anything you want to say about this before we move on to some of the questions in the chat?
Yeah, sure. So I just maybe I just want to add that the presence of glial cytoplasmic inclusions in healthy people is actually quite rare. It does occur, and of course I cannot speak for all of the pathologists who have dedicated a large part of their research to this topic, but it is, I think it is more common in diseases like Alzheimer's, maybe even Parkinson's disease, that there is indeed that there are deposits of synuclein without the presence of actual symptoms. So clinically seemingly healthy people, but in MSA, this is not as often, or doesn't happen as often as in other diseases. Also, the burden and the load of synuclein that you'll find in brain material of people with MSA is much higher, sometimes even 10 times more higher than for instance what is observed in Parkinson's disease. So there's of course this and a lot of experimental data that suggests that there is an important role of for synuclein and synucleinopathy in the disease process and in the etiology of multiple system atrophy. And so there's of course a significant evidence that we should be looking at this protein, but of course we cannot neglect other clues or other avenues that we should be investigating, as well as Alberto just mentioned.
Okay. So there's one of the questions here. I was going to read it verbatim. Are MSAP and MSAC the same disease in that they both target the same areas of the brain, but they just do it in a different order?
They are defined by clinical features. How we diagnose this disease is based really on clinical features. We see someone with mostly cerebellar features at the bedside, and if we have no other good explanation for that, we refer to it as MSAC. The final diagnosis, of course, can be made only on autopsy. We don't have a test that can definitively tell us that somebody has MSAC, nor MSAP. So the paradigm here is that we clinicians get to tell patients, you have MSAC or MSAP, trust us, we're the expert. And we are the only field of medicine that get away with this. In other fields of medicine, that doesn't happen. There are suggestions of a disease that a clinician could say, you have something that looks like breast cancer, but I have no idea what it means to you. I would have to do this test, this other test, this other one, and then I'll know what disease you have. And then I'll know how to treat you. We skip all of that. We say, trust me, in my experience, and I'm an expert, you have MSAP. I cannot prove it to you, but trust me, I've generated this information from my forebears learning on many autopsies. And I know that if I were to do an autopsy on you today, I would confirm that I'm right.
But isn't it actually true that MSAP and MSAC do affect actually different areas of the brain?
So the distribution changes, right? That's part of how MSAC might be conceived as different as MSAP. But the point that I'm trying to make is that we're making a diagnosis of a disease based on how it looks to us, a flawed clinician such as myself. Now, I think very highly of myself, but I don't think that I can really define what a biological entity is. Now, my forebears had nothing else at their disposal than a microscope and waiting until somebody dies to make sense of what happened during life. That was in the early 1900s and may have made some sense in the 20th century, but it cannot make more sense or continue to make sense in the 21st. We're using technologies.
It's actually true. There are a lot of diseases that are made, a diagnosis is made only on clinical presentation. It's not like it's only MSA.
Oh, most of them are made on clinical diagnosis, most of them. No, MSA is no exception by any means. We tell people they have a variety of conditions because the diseases were defined at a time when all we had at our disposal for classification was autopsy. And all I'm saying is a part of my role with my colleagues is to start moving us into what's referred to as precision medicine. If we really want to make a dent and slow the progression of people affected with any condition, including MSA, P, and C, we have to at some point embrace the concept that it isn't up to us to define what diseases people have. We need to be agnostic and need to understand that not two individuals have the same disease, even if they have the same diagnosis. And I think most people who have experience with MSAP and MSAC, right, as you do, as many others do in this call, know that the experience is very different. And chances are that experience is different from one person to another because the underlying biology is different. Now, there are common denominators, and those common denominators are what we use to define the conditions. But if we define the conditions by what's common rather than what's different between people, then we will not be able to slow the progression of anyone.
Isn't that true also with any condition? That's why some people respond to one type of antihypertensive medication and other people don't. So isn't the precision medicine?
So let's keep the discussion. So there are two things here that are important to distinguish. You just mentioned antihypertensives. Now, we have two kinds of treatments. One are symptomatic treatments. We go after what's common in antihypertensive. The common thing is blood pressure. We try to reduce it. That's what's common. Do we know what's the cause of the hypertension as an individual? We don't. In some we do, but in many we don't. Now, in those we do, where that's coming from, then we target the disease. Right? Sometimes it's adrenal diseases of different kinds. Sometimes are endocrine diseases, pheochromocytoma. We treat that, and then what happens? The blood pressure normalizes without us having to continue using antihypertensives. In MSA, we use levodopa, we use amantadine, which we have symptomatic treatments. Yes, some people benefit, some people don't. What we don't have any of is disease-modifying treatments. And that's what this discussion is about. So I don't want to say we don't have anything to offer to MSA. We have plenty to offer, but we've never been able to slow anyone's progression because we've insisted we already know what MSA, P, and C are. And so we just need to figure out where the disease is coming from, what's causing it. Well, if we already know what the disease is, all we can uncover are common denominators, and common denominators at a population level cannot be that relevant at an individual level.
Okay. So there's a comment here in the box. Thank you for presenters for the work on MSA. As you know, research on MSA often piggybacks on the research of other neurodegenerative diseases. So thank you for those who have taken on the challenge of studying a rare disease. That kind of leads me to the question of how much, how much of a problem are we facing that we're relying on other disease processes to guide us in our research on MSA? I mean, there can be parallels, but there could be big differences with that. what are the challenges we might face in taking that approach? Let's start with Dr. Wouter.
That's a very valid point. I think much of what we know is from, or much that we know in terms of the pathogenesis of MSA actually comes from Parkinson's disease and related synucleinopathies. And in fact, some of the conceptual challenges that we're investigating also apply to other neurodegenerative diseases. So there is a lot that we learn from other diseases, and that and to some extent we can extrapolate to the research that we do for multiple system atrophy. And so in that sense, I think it's been very useful and very powerful, not just to look at one disease, but look at a spectrum of diseases. But then again, it's important not just to look at a class of diseases or a syndrome, because that might not us learn, or that might not learn us enough about the actual biological mechanisms that might also differentiate people with multiple system atrophy. So we need to look for specific processes. We need to look for why multiple system atrophy is so different between patients with MSA, but also between different syndromes and between different conditions, like for instance Parkinson's and dementia. And I feel like we're only starting to understand what those differences might be, and why multiple system atrophy is so unique as a disease, and why it's so distinct from those other diseases. But this is unfortunately, I think, only the beginning, and there's only so little that we know about MSA. So we need to keep doing what we're doing, and we need to keep pushing this research so that we understand what is going wrong, and so that we can find a good therapeutic strategy to intervene with those processes that start malfunctioning very early in the disease. But this is still a very open question, and still a lot, I think a lot has to be done in that regard.
Here's the next question. My husband had no infections prior to MSA diagnosis, but might his volvulus have been a trigger? And that relates to a question that I had regarding your presentation on UTIs. I mean, is there any reliable data on how many patients had UTIs prior to an MSA diagnosis? I was curious about that part of your research.
That's a question we also had, and so of course it's very difficult to specifically identify all those people that had a UTI, because very often they also go unnoticed before a diagnosis of MSA, and even after diagnosis of MSA. UTIs are very common, and I would say more. I think that they are more often, and they mostly go unnoticed, in fact, than people report their UTIs. So I think they do contribute significantly to the disease while the disease is already ongoing, but also before diagnosis. And so in the retrospective study that we've done with our colleagues in Denmark, where we looked at, I think it was more than 5 million people in Denmark, and we looked at all of those that had a registered UTI, and so we tried to correlate the UTIs with diagnosis of multiple system atrophy. And so there were definitely much more people with UTIs that then went on and developed multiple system atrophy. But obviously, of course, there were also people that had UTIs and did not develop multiple system atrophy. And so that portion significantly goes up for that group that developed multiple system atrophy. But also we noticed that for those people that developed or that received the diagnosis afterwards, that also UTIs were repeated and became much more chronic than those that weren't diagnosed with MSA. And so this all happens before diagnosis of the actual disease. So it's very hard to pinpoint how many people there are that will have a UTI because of registration and the way how these UTIs are recorded. But there's certainly a much larger portion of people that eventually will develop MSA compared to the general population.
And what about the person's specific question about volvulus? Could that have been a trigger?
I think that's a really interesting question, because so colon obstruction will also certainly lead to inflammation in the gut, and so the colon shares a large part of the sympathetic and the parasympathetic pathway with the urogenital system. And so the mechanism behind that might be quite similar. So there's inflammation in the colon, especially that lower portion of the gut, the GI tract, and an inflammation or a trigger might set up a similar mechanism that might lead to similar effects. And so we've been studying in UTIs because it's because one, we could work with clinical data that was documented, but also because of the experimental models that have already been optimized in the experimental models that we could work with. We could also do it, for instance, with other models, or we could also look at, for instance, gut inflammation or colon inflammation, or inflammation that happens at these sites. And in fact, I think some people have also done this, but then looking, for instance, at inflammatory bowel disease, IBD, and I think there also there was a link or an association with people that had IBD and then again diagnosis of multiple system atrophy, I think 5 to 10 years after the first reports of IBD. So there might be a similar mechanism. And so the UTIs we're studying as, so we're studying the UTIs as a potential trigger, but we think that this mechanism might also apply to other inflammatory triggers. And so that it's more of a concept that fits within this frame in which we are thinking about the onset of multiple system atrophy. So it could certainly apply to different types of inflammatory triggers.
And I don't want to, again, monopolize the questions here, but you did mention autoimmune diseases as well. And it's interesting, my mother had Sjogren's disease before her MSA diagnosis. So I was wondering, are there specific types of autoimmune diseases that have been more highly correlated with MSA diagnosis?
It's interesting you mentioned this, because I was going to give this as an example, because I believe there's also an association with Sjogren's disease and multiple system atrophy. I think this was a very small study, but I think the link was statistically powered enough and significant enough to actually establish a link. So there might certainly be an autoimmune component. How that would exactly work is it's hard to say, because there hasn't been much done in regarding any questions regarding autoimmunity and how that might affect multiple system atrophy. This is certainly the case for Parkinson's disease. So I would expect that maybe similar mechanisms might be involved in MSA as well. But currently we're just lacking studies and lacking the data just to corroborate or just to say anything meaningful about autoimmune disorders and multiple system atrophy. But I wouldn't be surprised if that would be the case. So this could be a possible facilitator that would facilitate any pathology or any development initiated by a certain trigger in the brain or outside of the brain, and then facilitating the progression or facilitating the development of multiple system atrophy.
It seems like we're just starting to get to understand the association with inflammation in general in the body. So I want to move on to the questions here. Upon autopsy, my husband was found to have both MSAP and MSAC. How common is this?
Most patients with MSAC go on to develop features of MSAP of some sort, but the predominant source of disability remains the cerebellar features, the ataxia, the incoordination. Similarly, people with MSAP develop features of MSAC because the underlying pathology is the same. It's just the distribution in the brain that changes from people to people.
Do you believe the MSA will eventually be found to be a hereditary disease?
Will there be individuals that would be genetic? Sure. If we think MSA isn't one disease, then there might be many expressions of it. Sjogren's could be one expression of it. I cannot imagine that if Sjogren ends up being connected to, let me say, epidemiological, meaning at a population study, that would somehow be applicable to everyone. Same thing with the herpes simplex virus. I saw a question about that, and yes, it's been demonstrated it causes something indistinguishable from Alzheimer's. Indistinguishable. People who aren't recovering well develop something that clinical features, and in fact on autopsy looks identical to Alzheimer's. Now, would we want to give everyone an antiviral treatment? No, because that's going to be explaining a very small percentage of patients. By and large, though, MSA is considered sporadic. 99.9% of patients have no history family history of it. So I wouldn't expect that if there is a genetic component that it will be inheritable, but there likely are genetic vulnerabilities, and we just don't know which those are in which individuals.
Yeah, maybe I could also add here, there's probably more evidence that there are no strong genetic risk factors, certainly no causative genes, and that there is more evidence that there are, either no or just very little risk factors present that might explain, why people get multiple system atrophy or not.
But isn't there a genetic association with having an autoimmune disease, even if a person doesn't have the same autoimmune disease as their parent, let's say? I mean, isn't it the case that if someone has some kind of autoimmune disorder, that their children are somewhat more likely to have an autoimmune disorder of some kind?
I believe that's correct. Yes, it's true. And it'd be fascinating to find whether that somehow triggers in some individuals a neurodegenerative condition. We now understand that there are certain antibodies that are associated with neurodegeneration. So it could be that an autoimmune family history or personal history could be the tip of the iceberg for widespread autoantibody development. And in some cases, some antibodies do create troubles in certain parts of the brains that could end up leading to something that's currently recognized as MSA, for instance, but it can be other conditions. There is an antibody that was identified a few years back called IGLON5 that leads to a type of Parkinsonism called progressive supranuclear palsy. Looks very similar to it. There is just a little bit more of sleep-related problems, and this would be one way to think about that form of PSP, progressive supranuclear palsy, as being autoimmune in nature. At the moment, we think that's rare, but before this was discovered, we never even thought about a connection between autoimmunity and neurodegeneration. So this would be perhaps the beginning of additional discoveries in that direction.
There's a question here. Have patients also shown inflammation in the liver? I do believe you mentioned that, Wouter, in your presentation. So this person's saying, I've been showing elevated ALT, AST in the past few months, non-alcoholic fatty liver and inflammation.
That's something I'm not well aware of. I would have to look that up, but it might. But I'm just now guessing here. So I would need to check the answer for that question and come back to it.
There was one thing I didn't understand maybe about the experiments you talked about. It sounded to me like what you were comparing was mice that didn't have any kind of problem, and then mice that you had genetically engineered to have higher accumulations in the brain, and then you injected them with a UTI. So I was trying to understand, that model. I guess it's the only way to do it because, what you're basically testing is whether you have people who kind of have higher accumulations in the brain, so they're going to be more prone to reaction to an infection. So it's not necessarily that you get an infection and that's going to trigger the accumulations. The accumulations are already kind of there, and the infection accelerates it. Is that how I should be understanding the experiments you described?
Yeah, and it might also be a chicken or the egg question.
Yeah, that was the question.
So right, exactly. So there were a couple of paradigms that we were testing. In one paradigm, we were looking at how, let's say, a normal organism responds to UTIs, and then in another paradigm, we were testing how these mice that were already accumulating alpha-synuclein in oligodendrocytes, how these were responding to UTIs. And so our expectations were that in the normal mice, we wouldn't see any pathology or we wouldn't see any brain damage. And that in the mice where there was already a bit of oligodendroglial alpha-synuclein, that we would see an exacerbation of pathology and that these mice will develop a phenotype that would resemble that of multiple system atrophy. And so, in fact, in the second case, this indeed happened. So these animals that were accumulating just a tiny bit of alpha-synuclein, but not sufficient to actually cause pathology and to cause aggregation of synuclein. So these mice just continued and developed more pathology much faster than, let's say, their litter mates or other mice that were treated in the same paradigm, but with saline or with PBS or just with any control condition that does not result into infection. So the infection was gravely aggravating and accelerating the phenotype, and was causing much more MSA-like features than the other animals that were treated with a non-infection. And what did actually surprise us is that when we treated also the normal animals, so the normal condition, that also in these mice we were seeing oligodendroglial synuclein expression. And this is something we didn't actually expect. And so these, what we call wild type animals, normal mice, when they received a UTI, we also saw in the brain, again, accumulation of this potentially pathogenic protein within these cells that, again, are so badly affected in multiple system atrophy. So, what it means, I think what it means is that these UTIs can have different consequences depending on their timing and depending when they happen, and the consequences might get worse while these UTIs reoccur and when they become chronic. So that's one, I think, important conclusion. I think the other conclusion and the other, let's say, unexpected fact we saw is that these animals start to phenoconvert. And so what that means is that whenever you give them an acute UTI, the immune system clears the bacteria, kills the bacteria, and the infection stops. When the disease is starting to progress, we see that the infection cannot be cleared anymore. And so there's pathology in the brain, in the central nervous system, which is far away, far outside of course, far away from the urinary bladder. But even when there's pathology happening inside the brain, this apparently starts to affect the urinary bladder as well, because these animals cannot clear the infection anymore. And anything that was acute just a couple of weeks ago now suddenly becomes chronic. So these mice cannot resolve the infection anymore. Bacteria keep on multiplying and the infection persists, keeps on happening until we treat them with antibiotics. So for some reason, there's a connection, or there's some connection that not only goes from the bladder to the brain, but also apparently from the brain to the bladder. And again, the reasons behind this are a bit elusive. I mean, we just started to look into why this might be. And there's a lot of evidence from other studies looking at multiple sclerosis or Alzheimer's or just simply aging, where now groups are seeing that oligodendrocytes can actually adapt a disease-associated state that is very specific for inflammatory triggers. And so these oligodendrocytes start expressing proteins that are typical of an immune response. And with that, they also start expressing, very interestingly, alpha-synuclein. And so there's a subset of these cells, disease-associated oligodendrocytes, that start functioning very differently compared to other cells in the central nervous system. And we think maybe that because of this conversion, this will affect also more, this will also have a more systemic effect, and that this will cause this phenoconversion that we are seeing, so that in our animal model, at least in our experimental model, we see this shift from an acute to a chronic phenotype. If that answers your question, yeah.
Yeah, so in the clinical session yesterday, there was a talk about the possibility of using skin biopsy in the diagnosis of MSA. I was wondering if you could explain to all of us, like, what exactly does alpha-synuclein do? If it's a presynaptic protein, and if it primarily is there in the brain, how does it get to the skin?
So it's endogenously expressed in every neuron in our body. So it's an incredibly abundant protein. And so we know synuclein mostly because of its role in the central nervous system and inside the brain, but it's also expressed in the periphery. So there's a lot of synuclein in everybody. In the gut, there's literally, I think, milligrams, even grams of synuclein in the gut, in the appendix, in the colon, in the urinary bladder. We also see a lot of aggregated alpha-synuclein even in healthy people, but also more in people with multiple system atrophy. There's also synuclein in the skin, in the esophagus. So it's really throughout the entire body. So you can really find it anywhere. So it really facilitates, or it helps with communication between neurons. It's enriched in the synaptic terminals of neurons, and so this is where the protein is normally endogenously expressed. So you'll find it throughout the body. But with synucleinopathies, you can actually see that the protein starts to adapt a different conformation, and that it, therefore we think might become pathogenic. And so these inclusions, or these aggregates of alpha-synuclein, then start to form not just in the brain, but also outside of the brain. What they're doing outside of the brain, and what the role of these inclusions might be there, is still a bit elusive, but it seems that it can be used as a diagnostic marker, because it seems to be that there is some specificity there as to in which people these proteins starts to accumulate, and in which people it doesn't.
There's a great question here about brain banks. Regarding donation to brain banks, are the tissues shared among research centers? I've seen a few brain banks that only accept donations from individuals who are patients. This seems to result in fewer tissues available for research, and sort of speaks to the broader issue with research these days in terms of payment firewalls and not really the free exchange of information that science is grounded on.
I agree. I just want to comment briefly on this issue of synuclein in skin, and the fact that, as Wouter pointed out, synuclein is everywhere in the body, almost everywhere. You just happen to think of it more as if it were a brain-only protein. But part of the reason to rethink a bit of our toxic synuclein approach is that synuclein is everywhere. So we're born with lots of it. The gene that codes for synuclein, SCNA, is being preserved in prehistoric genomes, and evolution made synuclein redundant. Not only do we have alpha-synuclein, but we have beta-synuclein and gamma-synuclein. So evolution has put us with lots of synuclein. And the question that we have to pose ourselves is, do we think that evolutionary forces were at play so that the minute our brains will be fighting against anything, an immune dysregulation, an UTI, whatever it was, that we would then have our own built-in toxin? Or would it rather be that synuclein is so important, and in fact, it's been demonstrated to be so good against infections, it's part of the immune response, that the reason we have so much of it is because we have to navigate a world where there are many insults as we age, more of them, some of which are infectious, some of which are immune dysregulation, et cetera, such that then we would actually have a first line of defense. And I wonder that that's more likely the reason that synuclein is very important. And Wouter and I participated in the Synuclein 2022 meeting, and to your point, and you ask about what does synuclein does, right? And actually this meeting, the whole first day of the meeting was spent on understanding what is it that synuclein does. And it does a lot. It's a whole day for it. And there are many things that we think it does, but we just don't know about it. And of course, many things we don't know yet, it may never know. But it appears as if synuclein is incredibly important of a protein. That was the first day of the Synuclein 2022 meeting that we have. Now, the second day is spent beginning to think how to get rid of it. So we go from synuclein is so important, it does so much, is evolutionary preserved for so long, to let's try to figure out how to get rid of it. And so it's as if we're missing a day in between these two, where it says, what happens once we no longer have the normal function of the protein, because the protein becomes entrapped in these pathologies, in these Lewy bodies or in these oligodendroglia inclusions, etc. What happens once a protein can no longer function? That's the part that we're missing. And so I wanted to just point that out as food for thought.
Okay. And what about the question that the participant asked about brain banks and sharing?
Yeah, I agree. So part of the other part of that answer to that question, which is an important one, is that there are very few resources within our institutions to deal with brain donation. So we are we always encourage our patients for brain donation, but there is an expense around the brain harvesting, the processing, the storage, the analysis. All of that comes with a cost. So we offer it to our patients, and we've had a good success in bringing them into our program, but we cannot take patients from that we've never seen, because part of the process is to ensure that we have a lot of information about them while they were alive, so that then we can make correlations between what we found in autopsy and what kind of clinical features they had when they were alive. So that's part of the issue why it isn't. Now, if we had unlimited resources, this would be different. And so part of it is just that there aren't many resources allocated to brain banks. Honestly, brain pathologists aren't that many in the world. Unfortunately, this is a scarce commodity. It's wonderful to have people like Jeff Cordauer, for instance. He's been doing this for a while, but he is a rare species. We don't have that many people like him working on this. So that's kind of it. Now, as I've alluded in my presentation, we have to try to make sense of what happens to people while they are alive. We cannot continue thinking that the clues to understanding them lies in their brains once they are dead. I wish to believe that we have a lot more to offer to people before they die. Now, I appreciate greatly my patients saying, when I die, I want you to have my brain. That's important. I love that. I think that's important, but we really need to move in the direction of saying, what can we do to learn about brain diseases without a brain autopsy?
Well, and how many research studies are currently actively recruiting living patients?
We have a biomarker program that's recruiting people with MSA, as well as a variety of other conditions, but MSA-C, MSA-P, we definitely are. Now, within that study, we have patients to sign up for the optional brain bank program. So that's one such program. Now, there aren't that many out there. And so I think the other issues, of course, which studies are recruiting for trials, that's a different category. It's increasing, but not quite yet at the level that perhaps it should be.
And I was just asked to remind people that Defeat MSA does have a brain donation page. So if you're interested in that, please check that out. There was one other question here before I forget. And this is for Wouter. In your studies, was it a matter of months that MSA symptoms were recognized after the UTI was introduced to the mice models?
I think it was about 9 months or 9 months when we saw the first symptoms. So it started with urinary symptoms, and then we also started noticing behavioral deficits afterwards. This is, I mean, of course, this is relatively short, and this is, of course, an experimental model. So it's difficult to predict what would happen in a real, in a real situation. But for our epidemiological work, we've looked at a time frame of 2 and 4 years and 4 to 8 years before diagnosis, in which we believe a trigger might result in symptoms. So we think that the prodrome of multiple system atrophy would take somewhere between probably around 4 years on average. So we've looked for anything around that time, let's say between the trigger and the disease onset. And so in an experimental animal model, of course, this will go much faster.
I want to just go back really quick to the question of the brain banks. Given the cost involved, aren't you getting a biased sample too then of people? Like maybe you're not getting as many people from low income, low resource areas, and that could have implication in terms of a patient's overall health. So how do you account for that in the brain banks that are, or the brains that are donated to these brain banks?
I agree. I think that's an issue. Yeah, it's a very biased sample. That's one of the problems when interpreting a lot of the data that emerges from brain banks, that you have to be mindful. This is only a slice of the MSA pie, and how much does it tells us about the whole? Very hard, but the ways to go around it are going to require governmental changes to increase funding for rare conditions such as MSA, and ensure that brain banks get to be better endowed. There is an endowment that really needs to happen, not just one-time funding, but you have to endow them for ever, right? Because if let's say you had a funding for 5 years, well, what's going to happen if that doesn't get renewed at the end of it? What do you do with this brain, right? All the costs of maintenance and so forth. That's why this is really a long-term commitment, and it's hard to fund it through research grants that have a finite period of time.
This relates to Dr. Kordauer's presentation, but it also relates to the presentation, the other two presentations on the research studies. Can you explain the difference between the PLP, MBP, and CNP promoter models? And I know that at least one of the studies that was mentioned relied on the PLP model, but it seemed like that was a better fit for Parkinson's disease. So what's the difference between those three types of models?
Yeah, I think I can answer that question. So the difference between those models is that they're based on a different type of transgenesis, and so the PLP and the MBP and the CNPA's model are using different, let's say, promoters, transcription factors to drive the expression of alpha-synuclein in oligodendrocytes. So in the end, they're all accumulating. These mice are all accumulating alpha-synuclein in oligodendrocytes, and the only difference really between those models is that there's a different promoter, a different transcriptional regulation of alpha-synuclein that is driving the expression of synuclein. So probably the levels are within the expression level of alpha-synuclein, and maybe also between the regions where synuclein is expressed. So in the end, they're all overexpressing alpha-synuclein in oligos. But it's probably just more about the expression levels and the pattern of expression that is different between these animal models.
One other question that I had when I was watching your presentation, are chronic UTIs associated more with MSAP or MSAC? Is there any kind of bias towards either one of those? And I think a general question I have is, where do we stand on research on MSAC? I mean, because of our, I don't want to say bias, but because we rely so much on other disease processes and we know so much more about Parkinson's disease, it seems like MSAC is not getting as much attention. So first, is there an association between UTIs and one or the other, and where do we stand in our research?
Yeah, absolutely true. I think MSAC is still being under-recognized, especially from the experimental part. And I think much of that is because of historical reasons, because of all of the animal models that are now being transferred and being adapted from Parkinson's disease to multiple system atrophy. And so in that sense, it makes more sense to actually start with an MSAP model instead of switching immediately to an MSAC model. As to the to your other question, I think the infection model that we're using might actually more closely resemble an MSAC phenotype than an MSAP phenotype because of the anatomical organization and the way how the disease might progress and spread throughout the central nervous system. We would hypothesize that the brainstem and the pons and the cerebellum would be the first regions to be affected after a trigger would occur. And in that sense, for MSAP, we would expect a totally different trigger to cause MSAP if a trigger would be involved in MSAP, of course. So it really gets back to, again, the different subtypes, the way how these diseases might originate, and again, the probably different biological mechanisms that are involved in these diseases. So we're talking about multiple system atrophy, but there might be many different forms of multiple system atrophy, which then again will also require different therapeutic strategies to intervene and to stop the progression of these different types of MSA.
And we only have 2 minutes left, so I think if we can give 1 minute to each of you to talk about what's next in your research plans.
For 1 minute, gosh. So I think it's something that came up quite frequently now, is that again there's not much, there's so little that we know about multiple system atrophy, and there are a couple of really interesting avenues that we should look further into. Of course, there's I think animal models, they have their flaws, but they're also really important in understanding what is happening in MSA as well. So from the experimental side, I think we really need to continue the way how we're currently doing our research, and we need to keep building on these findings. And so I think it's really important that we just keep continuing in this way, and again keep expanding, try to understand again what the role of the immune system is in all of this. We need to try to understand these facilitators. We need to try to understand again what these different biological mechanisms are that are involved in multiple system atrophy, how we can target them, how we can treat them, and then how we can stop multiple system atrophy.
From our perspective, we're very interested to move away from understanding diseases to understanding people with diseases, on the recognition that if we then can accept the fact that not two individuals are alike, we are conducting research acting as if we mean it. So we're trying to, in fact, create a platform through our biomarker program that allows us to recognize an individual's biology with bioassays developed to determine if any of them are biological candidates to therapies that already exist that can be repurposed. Over the last 4 decades, we developed great therapies that have interesting mechanisms of actions, all of them invariably negative in clinical trials. Those therapies have a lot of value, and we just never knew who the right candidates for them are, and that's what we're creating the platform for. That's our CCBP study, the biomarker program I alluded to earlier. And then separately, we're moving toward the preclinical studies needed for protein replacement strategies, recognizing that all of these diseases are protein loss disorders. We're trying to figure out ways of how to replenish the types of proteins, such as synuclein, that are being lost in the process. That could be a collective approach. Precision medicine, of course, that's required to understand what is at an individual level biologically relevant that we can correct.
All right, well, thank you very much. I appreciate both the speakers for being here. Everybody else, if you want to attend the patient and caregiver breakout, please feel free to stay on the line. Otherwise, have a nice afternoon, everyone, and thank you, or morning, or wherever you are, and thank you for participating.